In this video, we continue building our foundation for chemical biology by reviewing DNA and RNA, otherwise known as the nucleic acids. What we are going to do specifically in this video is look at the building blocks of DNA and RNA. So in order to do that and get started, I'm going to go ahead and share my screen. There is a handout that goes along with this video, which you can access under this particular module, right with the link for this YouTube video. You'll see a link to this handout that I'm going to display. Additionally, you can also go under files and find it. It will be listed under CHM4930 and listed as the nucleic acid handout for module 1. So just as a reminder, a common theme that we have seen throughout our time talking about issues related to the biomolecules that we're focusing on is that we have monomeric units that join together to form complex biomolecules. For example, in the list that we see here in the upper left-hand corner of the screen, we have our amino acids linked together via those peptide bonds or amide linkages to give proteins, complex biomolecules comprised of hundreds to thousands of those amino acids. Monosaccharides were our individual sugar units, such as glucose, that existed as the open chain form and the cyclic form. And particularly when those are in the cyclic form, they link together to form our complex carbohydrate molecules, such as cellulose, such as glycogen, and other carbohydrates that play essential roles in cells and essential roles in our bodies as well. Monomeric lipids were compounds such as fatty acids that ultimately are assembled into the more complex lipids, such as steroids, such as phospholipids that make up the cell membrane of a cell. And now, as we get into our final group of biomolecules, we analogously recognize nucleotides as the building blocks of our nucleic acids. And we're going to focus primarily on DNA and RNA as being the nucleic acids of interest. So when we are looking at nucleotides and thinking about the chemistry of nucleotides, the general chemistry of a nucleotide, as an overview, we can see in the lower right-hand corner of this slide, what a nucleotide consists of is a nitrogenous base. That is this portion here in the upper right-hand corner of that structure that I just highlighted in blue. And generally speaking, DNA will contain four different possible nitrogenous bases incorporated into these nucleotides that would ultimately be assembled into DNA. Those are adenine as one of the bases that can be found in DNA, and I'm just going to annotate that as DNA to indicate that that nucleotide adenine is one of the nitrogenous bases that we could plug in here into a nucleotide that ultimately gets assembled into DNA. Additionally, guanine is another nucleotide that we would find in DNA. I don't expect you to memorize these structures because you will be able to look this information up on quizzes in this class. And then cytosine, also one of the nitrogenous bases, and thymine. So those are the four unique bases that are found in DNA. And what we are going to see is that these nucleotides are going to covalently bond, as we will see in the next segment. And we'll have long strands of those that then engage in hydrogen bonding with complementary strands, and we see a diagram of that here that I'm highlighting hydrogen bonds linking two strands of DNA. The strand on the left that I'm highlighting here, hydrogen bond interacting with the strand on the right to make this structure relatively stable. So the four nucleotides that we will see appearing in DNA are abbreviated as A, G, T, and C to emphasize that the difference between those nucleotides as the building blocks of DNA are the structure of the nitrogenous base. What all of the nucleotides will have in common is they all have a sugar here, specifically this 5-atom ring sugar that I'm circling. And it differs a bit between the sugar that is found in DNA, deoxyribonucleic acid, and the sugar that is found in RNA, ribonucleic acid. So in DNA, the sugar molecule that is found here is a 2' deoxyribofuranose, is what we would find as the structure of the sugars that are found in DNA. And we can dissect that name based on what we know from our earlier discussion of carbohydrate chemistry. Furanose refers to cyclic sugars that have 5 atoms in the ring. And we see that indeed, this fits that theme because our sugar has 5 atoms in the ring here, our ribose. The ribo part of that is referring to the specific sugar that is here, as well as the deoxy part. The deoxy part indicates that there is no hydroxy group at carbon number 2 of the ring. And carbon number 2 of the ring we can find by using numbering similar to what we used when looking at carbohydrate chemistry and carbohydrate numbering. We start by numbering at the anomeric carbon. That is the carbon that was originally part of an hemiacetal group. It's the carbon atom that links to the ether group within the ring, which I'm highlighting here with my red laser pointer, and then also connects to originally the hydroxy group of the anomeric carbon or the hemiacetal group. And now when we bond between the sugar and the nitrogen in space, we have a direct link between carbon and nitrogen there. So the carbon oxygen bond that would have originally been here on the ribose before it underwent this reaction with the nitrogen in space has been replaced with a carbon nitrogen bond. So this is position 1 because that is what originated as the anomeric carbon. It originated as the hemiacetal group of the sugar molecule that is the precursor to this. And then we go around the ring, 1, 2, 3, 4. And so when we say that it's 2' deoxy, what that means is that in the nucleotides that are the building blocks of DNA, there's no hydroxy group at this carbon number 2 of the ring. And that's why it's called 2' deoxyribofuranose. It's a ribofuranose because it's that 5 carbon sugar, ribose. And it's deoxy because the ribose has lost its alcohol functional group at position 2. On the other hand, in contrast to this, if rather than looking at DNA, if instead we were looking at RNA for contrast, what is present there rather than the 2' deoxyribofuranose is just ribofuranose, which will have a hydroxy group at that 2 position, at carbon number 2 of the ring. So in the case of looking at the RNA, ribonucleic acid, there is a hydroxy group at carbon number 2 in RNA. So that's one of the differences between RNA and DNA is the building block composition. DNA has the deoxy group, and RNA instead in its building blocks has a hydroxy group at position 2. It is not deoxygenated. Another difference between DNA and RNA is DNA, as we mentioned previously, includes 4 of the building blocks, 4 of the bases that fill in here as our nitrogenous base in the lower left corner here, A, T, G, C. On the other hand, RNA extends that a bit and uses somewhat different bases. So in terms of what is possible for RNA, you will see adenine as a base, you will see guanine, you will see cytosine, You will also see uracil, this structure here in the middle right, which is not one of the ones found in DNA. And you won't find thymine as a base there. So thymine is unique because it is not found in RNA. So when we're thinking about the building blocks of DNA versus RNA, so thinking about DNA versus RNA from this building block perspective, to summarize, DNA has a different sugar group. It's going to have 2' deoxy ribofuranose versus RNA has ribofuranose. And they will have, to some extent, different bases that are found. So there is some overlap between the bases that are found in RNA and DNA, but there's also some differences. And I should add here that of these nitrogenous bases that I put in the upper right-hand corner, RNA also has some additional modifications of these structures to make them unique to specific organisms. They will have some slight structural modifications to some of these structures. So RNA incorporates a few extra nitrogenous bases as well. But the main things to keep in mind are that there are some differences in the bases that are used and incorporated into the nucleotides for DNA versus RNA, and DNA has a chemical structure difference in the sugar group that is present there. They all have in common the 3 overall general components that the building blocks of DNA and RNA have a nitrogenous base of some sort, a ribose-derived sugar molecule of some sort, and this phosphate group that is necessary for applications of this. So with that, what we're going to do in the next video is look at how we form bonds between these individual building blocks, these individual nucleotides, to link together into the primary structure of DNA or the primary structure of RNA. How do we form bonds between these to form a strand of DNA? And how does that strand of DNA interact with other DNA molecules? So we will get into that in the next segment here.